HIV life cycle begins with attachment, where the viral particle binds to a CD4 receptor on its host cell. Specifically, the GP120 envelope protein found in the ENV gene binds to CD4 and other co-receptors such as CCR5 and CXCR4 on T cells.
In order to enter the host, the HIV virus fuses its viral membrane with the cell's membrane. First, GP41 fuses the membranes, then viral enzymes and RNA inject themselves into the cells.
Reverse transcriptase converts viral RNA into double-stranded RNA, which can now be replicated. Mutations are introduced when new DNA strands are synthesized. This process results in a hybrid molecule, and RNA strands are also eventually degraded.
During the next stage, integration of provirus, newly formed HIV DNA enters the host cell's nucleus. An HIV enzyme called integrase hides the HIV's DNA within the host cell's DNA. Integrase cuts the host cell and pastes in the virus, a process that is specific to retroviruses. The integrated DNA is called the provirus, and this new gene is permanently inherited and can never be removed.
The cell then produces mature virions which can infect other cells. The first step in this process is transcription. During transcription, RNA is copied multiple times. Some copies serve as templates to make new proteins, while other copies provide the genome for viral replication.
Next is translation, where the provirus uses a host enzyme called RNA polymerase to create copies of the HIV genomic material, as well as shorter strands of RNA called messenger RNA.
Next is protease processing, where protease cuts larger proteins into smaller ones with varied functions. The POL gene makes enzymes for the cell, whereas the GAG gene makes 4 specific proteins.
The GAG proteins then package the genome, providing a structural shell. As the virus exits the cell, GP120, GP41, and other proteins help it escape. The virus also steals part of the cell's outer envelope. This envelope, which acts as a covering, is studded with protein-sugar combinations called HIV glycoproteins.
These HIV glycoproteins allow the virus to continue attacking other host cells.